Welcome, science lovers. Welcome to Science Sunday in 2021, January 3rd. Glad to be here, glad to see you all, virtually at least, and talk more about science. So thanks so much for joining in, either now on Science Sunday time, or as you can always get back to these as they're libraried and kept in the either the Humanists, Prairie State Humanists login area, or on the Page Unitarian Universalist Church site, and checking out our videos. So thanks so much for being here. I think I just saw Scott Thompson pull into the driveway, so he's coming in with his mask on in just a minute, and we're going live. So again, welcome to 2021. We've gotten through 2020, and with all the pains and challenges and so on, and we're happy to be here, and we hope you have a wonderful 2021, and we hope to be a part of it. A couple of things. We always are thankful to the DuPage Unitarian Universalist Church for supporting us here, and the AV team that's up top, providing this streaming service out to you. And I had a concept. I know the church is allowing up to 10 people at a time to come in if some concerns are taken care of, if things are done carefully. And so I had a concept of, if people wanted to check in, my thought was, and there's Scott. Hello, Scott. Hey there. If people are interested, maybe we'd like to do this is a new for you too, maybe, Scott. I thought we allow 5 to 7 people or so in the Science Sunday, and we could have a studio audience possibly.
Oh, yeah?
If people would be interested and respond to an email to me, and I'd say that first 5 people, I'll go to bat and try to see if you're interested in coming into the church and participating in Science Sunday from now on. We'll look and see how we can accommodate that.
Well, we got 3 very distinct sections here. We could probably get, about 5 people in here and have them very far apart. So.
Right, there's a lot of room in here, and it would be fun talking directly to some people too, rather than each other. I probably would be standing less on my left shoulder or left elbow and looking over towards Scott as we talk about things. So hopefully, if you're interested and want to participate and be our studio audience, and that could be, please send me an email, and I'll take them first come, first serve. If you're interested, and so, Happy New Year to you.
Happy New Year to you, and to all of our Science Sunday friends.
Yes. So we wanted to talk about vaccines today. I know that in 2 weeks, do you have your schedule, what you're planning on talking about?
Yeah, I was going to talk about some content related to political science and economics, specifically drawing from primarily 2 sources, 2 books that have recently been released about the idea of merit and the role merit plays in our society. So should be an interesting talk, I hope, and a deviation from our typical topics of biology and astrophysics.
More hard science, yeah. Also, that's the 17th of January.
Yep.
Right here at noon. And I just got asked by our friend Steve Nats, Dr. Steve, to interview him for the show that's coming up. He's having an opening of his show, of his book, an opening party, and it's called The Wisdom of the Flock. I talked about it a little bit, and I've read it with him. This is, I believe, his advanced reader copy, and I may be a reader. I'm not sure if I'm advanced, but...
Well, I don't know. I think you're pretty advanced, Mike.
So, talking about Franklin and mesmerism in Paris, and it's a historical fiction, and he's asked me to interview him as part of his book opening rollout party. So I've been preparing some questions and some thoughts about that, and I think he's going to seed me with a few questions to ask him about it and the way he wants to go. But it's an interesting mixture of science and history and a little romance, and and also a little bit of talk about science, history, romance. That kind of covers quite a bit of it, I guess. But why Franklin was in Paris at the time, and so it's been an interesting read.
I think it was because of a boat.
A boat.
Yeah, a boat took him there.
Yeah, the planes were all
We're all grounded back then.
grounded. Yeah, they
Yeah,
was,
that airport, the great airport that in the Revolutionary War.
right.
Forget which one that was.
Yeah, we had control of most of them, I hear.
Yeah.
So, excuse me. Without further ado, I know there's been a lot of talk and a lot of hope and a lot of interest placed in vaccines and vaccination, and so I wanted to start a talk about that. I do it with a lot of concern and trepidation. First and most importantly, I want to say that today's talk is a layman's discussion of the topic. It's a presentation of a serious issue of the day, obviously. I'm not a doctor or a credentialed scientist and therefore have a limited scope of expertise, if any, concerning the topic at hand. I ask you not to base any of your healthcare choices on my discussion today. Consult medical professionals to understand how to apply what we talk about today to your particular case. This talk is general in nature and may hopefully give you a broader knowledge of vaccinations and the COVID-19 vaccines, but what we say may not apply in your specific situation. I think I've covered myself pretty well.
That sounds pretty good to me.
Yeah, okay.
Yep. We do our best, but we're not, we're not licensed to give medical advice.
Right. So remind me to say that a few more times in here, too,
Okay.
at appropriate times, Scott. So, a fun fact, vaccines and vaccination, the term comes from the Latin word vacca. Are you familiar with what that is?
No, I'm not.
Vacca means cow.
Oh,
So Louis Pasteur is a great early microbiologist we'll talk about a little bit earlier, further on in the story. He came up with the name, actually, to honor Edward Jenner for his contributions to science. So we'll talk about why that is in a moment, but vaccines actually have a lot more to do with chickens and eggs and nowadays than they do with cows.
do you know which came first?
The chickens were coming after eggs. There were eggs way before there were chickens.
Okay, good to know. Thank you.
Yeah. So, at the time, we think about what we're going through right now. This pandemic is a problem, and it obviously is. It's a serious situation. But medicine over the last numerous decades and past hundreds of years, past centuries, has really developed a lot of means to try to keep us healthy. And it's thanks to science and scientific-minded people that I think we're in the positions of being able to overcome a lot of these challenges and problems right now. Smallpox at the time that it came out, it was a horrendous disease. Something like 30% of the people that caught it died. Many people didn't know why. They had no idea what caused it, what to do to stay away from it. It came in waves. For a while, it came heavily in the mid-summer and late fall or early fall, and then it seemed to change a bit, and then it was coming decades later. It was coming in the late fall and early winter, and people didn't know why that was or what was going on. So people were observant at the time and looked at things and saw some things, but it's an interesting. I have a bit of a timeline to show some of the things about vaccinations, timelines of significant developments. We have a nice screen in front of us that we can look at and see. It's a little far away, and it doesn't have the colors as well. It's put
Maybe
on
our AV helper upstairs can make this slide the exclusive focus for the moment. We
Yeah.
can zoom in on it.
Right.
Thank you.
So in the 1550s, smallpox was recorded. First known dates of inoculation for smallpox in China. Smallpox had been around for quite a while before that, but actually they were medically inducing weaker cases of smallpox in people in the 1550s in China. So their medical expertise was beyond what we were able to understand it at the time. They realized it came from something that was in the body and was able to be transmitted to other people. So they took small doses of it, and they called this, we call it in Western culture, variolation. They took the varioles, the pustules that were there in people that had smallpox, and gave it to other people in the hope that they would get a mild case and would then not have it again. Smallpox is known to not repeat itself. The body, as we know now, develops antibodies. So people saw that if you got it once, you didn't get it again. So smallpox, people were inoculated in China in the 1550s. Going on 1796, and actually, as you go on further from here, there seems to be about an 80-year or so development of things happening in terms of this timeline. So the spaces after this one are fairly even. Edward Jenner develops and tests the first vaccine in Berkeley, Gloucestershire, in the UK. He was a fairly well-trained doctor at the time and knowledgeable. This is in the time, obviously, right around this time, the American Revolution had been over for a short while. George Washington died just around this time by treatment for a cold that he developed while going out riding on a wet, rainy day in Virginia. And the common treatments at those times were things like bleeding people. I think they said George Washington got bled 3 or 4 times, in which they take significant amounts of blood to get the bad humors out of people's bodies, and weakened him significantly, and he probably should have lived a lot longer. He was a very strong man. But
Talking about humors, if you don't mind, the humors, the idea goes back, I think, to Galen, or even earlier in ancient medical practices, and there was the idea that there were these good and bad humors that course through your body. Red humor was blood and whatnot. Bile was another. But there's this idea of a black humor that would make you sick.
Right.
And medical science never validated these archaic claims about humors in your body, especially the black humors. So.
And as I've been reading about vaccines, I've seen some terms nowadays that are used, humoristic, I think, something about, and they still talk about body fluids or humors. So they still talk about the humors of the body, but not in the same concept and ways. But so at the same time that people were mistreating people for small colds and so on, for fevers and aches and dispositions that were not up to par, at the same time, Jenner looked and he noticed, and it was pretty well known at the time. He didn't make a great discovery. I think sometimes his discovery is overplayed. People were very knowledgeable about the fact that cow maids never really got smallpox, and they developed this immunity to it, as we call it nowadays. And
I've never seen a sick cow maid.
And they speak highly of you, too.
Oh, good.
Yeah. And so at that time, other people had actually tried doing some of these things, but Jenner was the first one that really made it a scientific study, that he tested and hypothesized and came up with means of doing it. Other people were kind of scratching around in the dirt, so to speak, of how to do it. So Jenner was the first applying scientific thoughts and methods to it. And he basically, I'll come back to this slide, but he basically vaccinated on May 14th of 1796. He kept dates and times and amounts and so on. A small, I believe, 8 or 9-year-old boy, James Phipps, with the vaccine, or with the pustules, with the fluid taken from a cow maid who had a mild case or had a case of smallpox, which was not as serious. It caused some marks, but it didn't cause death, didn't threaten.
You talk
to
about
cause
cowpox or smallpox?
Cowpox.
Yeah. She had cowpox, right?
Right.
Yeah.
So here's a graphic of it. Sarah Nelms, a milkmaid, was infected with cowpox, naturally, by milking cows that had the cowpox that specified or shown, delineated by the blue dots on here. So she had cowpox, and she was actively fighting cowpox, but it's something that doesn't really cause much problems. It's very easily overcome by the body's immune systems. Edward Jenner took some cowpox pus from Sarah Nelms and injected it into this James Phipps' arm. He scratched him more and then kind of just laid a little bead of it, almost like didn't have an inoculation or needles like they have nowadays. And Phipps fell ill with a mild case of cowpox. Then he recovered from that in a short order, and then scabs were collected from a smallpox patient, not Sarah Nelms, someone that actually had smallpox. And this is where Edward Jenner went a little bit further than most. He actually inoculated James Phipps with the scabs of smallpox, with the pus created from that, assuming that he wouldn't get sick and probably keeping his medical license and a lot of other
No lawsuits?
yeah, no lawsuits, nobody coming around with pitchforks at his door and bonfires and so on. But Phipps was unaffected. The protection was complete. He had been inoculated with the smallpox virus, yet did not present with any symptoms of it at all. His body was able to overcome it because it had been inoculated. It was immune to it. So that's kind of the process, again, showing him, a painting of him supposedly giving Phipps the injection of the cowpox. So that started out a lot of people thinking. One of the most famous was Louis Pasteur, who's done a lot for our milk in the last while. 1879, Louis Pasteur started a decades-long exploration of vaccinations for cholera, anthrax, rabies, and other diseases. All these were just devastating diseases in the late 1800s. You can see this is 80 years later, roughly. 85 years later, I guess, 83 years later. So he really admired what Jenner had done. It had not developed much more than that, and he started doing much more. He found all these different viral bodies and bacterial bodies, and he was able to develop ways of vaccinating people for a lot of diseases that had been so horrendous through the course of human history. He, again, like I said earlier, realized the importance of Edward Jenner, and it's pretty well known that he didn't have a lot of patience for people that were somewhat his compatriots, his competitors, let's say, people that were doing experimentation around the time. But
When you say he didn't have patience, you mean he got frustrated easily?
No, I think he didn't like their testing. He borrowed test results and changed. He did a few things that were...
Okay, I didn't know if you were talking about patience in the mood sense or patience in the people he was treating sense.
Right. He had a lot of antipathy towards people of his fellow scientists. That he didn't like their methods, or he didn't like their criticisms of his methods. But he liked Jenner because Jenner, by that time, was dead. And so he applauded Jenner's work, and he actually started the word vaccination. All his work, although you see not much of it had to do with cows. Anthrax was found in sheep's. Farmers in France at the time, if a sheep would die in the field, they would bury them in that field. And if they had anthrax, then it would get into the soil and the organisms would get into the grass, and the new sheep that lived in that same field would eat it, and the anthrax was passed along to the next one. So Pasteur recommended and convinced farmers at the time not to put sheep, not to bury the dead sheep in their field, and that was a big part of stopping anthrax. And he also went on with vaccinations and different things for that too. So you can see that not much of this has vaca cow involved in it after this point, but the beginning idea was from cowpox and the work that Edward Jenner did. For a while, it was very worried. People in England were very worried if you got injected with, and there were pictures of it. I didn't bother to put one up here, but cartoonists would draw pictures of after people got vaccinated, some would grow horns, and some would get lolling tongues, and some would...
That's how we got our first superheroes, I guess.
Right, yeah. So there were some concerns about what could happen by putting the bodily fluids of other animals into people. And again, that's the first kind of vaccine worries that have come out in the world, and they aren't all gone by now either.
Yeah, unfortunately.
So another almost 80 years later, Jonas Salk. Some of you may remember, I know I had a few childhood friends that were suffering the final throes of polio. But before I was born in 1955, the year that the Jonas Salk came out, after a 7-year development, he took 7 years of development to find out and to determine and to develop a method of vaccinating people against polio. And it was an amazing change for people. Polio was just this deadly thing that would strike from anywhere. No one knew where it could come from or how it could happen. So the scientific work that stopped polio's deadly reign is a fascinating story in itself. I don't have the time to talk about it now, but Jonas Salk and the development of that, and Louis Pasteur's stories, both of these are very, very interesting topics if you're interested in reading some, scientific history of how these things occurred. Just phenomenal.
Yeah, let's see. Yeah, several people, several scientists have been involved in this. Might have been some of the greatest heroes in human history for the number of lives they've saved.
Right. And that's, I think, how Pasteur viewed Jenner. The lives that were saved because of all these people's work are just phenomenal. Just inspiring story for young scientists out there to try to find the next thing to cure. If we can cure some of the other diseases coming around, and the COVID-19 story is pretty inspirational too. And if we look to it, another almost 80 years, 2020, COVID-19 vaccine, and vaccines really, I should have put an S there, are released under emergency authorization after 7-month developmental program, not 7 years like the polio vaccine. And we'll talk a little bit about why it's able to come out, what was done to prepare these things to get them to the point where 7 months, roughly a year after they know that this was a disease, that this was a problem, we've got a vaccine for it.
Yeah, amazing. And a whole new category of vaccines too, which
Right.
I'm sure you'll talk about.
Right. So significant developments. Here, just a short picture is a smallpox sufferer. In 1973, smallpox was determined to be obsolete after that time, completely eradicated. There are a few small vials of the virus kept, one in Russia and one in the United States at the Centers for Disease Control, just for medical purposes or study or to fund a lot of potential scary spy movies that allow the fact that maybe somebody will come in and steal them and start smallpox in. But a very horrendous look at the amount of pustules on this with smallpox. Like I said, 30% death rate from that. So I wanted to talk a little bit about vaccines, and some of this up till now has maybe been a lot of review for many of you, but also some interesting ways of looking at it. But this is some review of what goes on. This is from a short Nova article about immunity and vaccines and how they were developed, or how they actually work in the body. So I think you can get that up top and start it for us.
Immunity is a natural defense system of the human body. Imagine millions of immune cells, like white blood cells, all on the lookout for specific germs. If they spot something dangerous, like flu, they prepare to fight. When that flu bug enters your body, the white blood cells move in on it. The immune cells arm themselves and then replicate, creating an army of clones. Then they launch powerful germ-killing agents called antibodies, and they tag the germs for disposal. Once the germ is removed, the immune army can disband. But they leave behind memory cells. Their job is to remember the invader and to sound the alarm if it ever appears again. The first time around, it can take at least a week to mount the defense. But next time, the system is prepared. The whole process takes barely a day. It's so fast, we may never have time to even feel sick. A vaccine pre-arms the immune system by sending in a weakened or dead version of the germ, just enough to be recognized. The immune cells mount the defense, and again, you may feel the effect. But because the threat is low, the immune cells quickly disband. However, the all-important memory cells have been created. The immune system's now prepared for the real germ, but without causing full-scale disease.
Okay, I thought that was a fairly short and useful way of looking at it. Again, I don't think there's anything new to most people in there about it, but the memory cells are an interesting topic, and I think it gets a lot deeper than that, the types of cells that are created and how they're made and what's done with them. But, so that's a basic idea of what vaccines do and immunization. So a historical vaccine development leads to what I put up as these states of action. Some weakened or dead pathogens are presented to the body, usually by injection, but often also orally. The body's defense system recognizes this foreign invader and it activates, like it was shown in that video. White blood cells and other protective cells multiply quickly and produce antibodies. The weakened pathogen is then overcome, and the defense system settles down. It goes back to normal conditions, but creates memory cells that can respond more quickly than the next time the same or similar pathogens are present. So it's a pretty amazing structure that our body is able to do to not be infected more than once by some of these agents and to fight against these agents if they happen to come around again. And that's something that's obviously being counted on in this whole development of the COVID-19. Just as an interesting point, and I don't want to spend a lot of time on this, but these are some levels of a depiction of the cells of the immune system. There's the blood stem cells, which then can make all the other types of cells. There's a whole variety of, down at the bottom on the left side of your screen, or on the right side of your screen, I think you see the B cells and T cells and the killer T cells and so on. Those are all often generated in the lymph system and travel through the bloodstream as well too. So a whole variety of different types of cells that each have their own purpose. We don't have time today to get into the depths of each of those purposes, but that's an interesting topic as well too. So wanted to just see that and have you see that.
So it's pretty complicated.
Yeah. And then a miracle happens.
Oh, no.
So how do the COVID-19 vaccines work though? And the key to all this is that we're not using this historical technique of weakening a pathogen and then putting it into the body. The same thing Jenner did and the ancient Chinese did and that Louis Pasteur did. We're not doing that. We're using an inoculation technique, a vaccination technique, that has some differences to it, and those differences are valuable in production methods, in timing, in efficacy, in also just the speed with which things can be done.
And safety.
And safety as well, too. You're not putting a weakened pathogen or any pathogen at all into a body. So these are all good things. There's still a lot to be done. This is, the beginning of science, and there's always side effects that could possibly come out of this, and there's things to look at and study at, and people will be and should be doing it for years. But there are a lot of hurdles that are cleared by this, and just the long-term side effects are something that can be investigated as time goes by. But the history of science shows that you can count on things that have happened in the past that worked well as happening as well here too. So the key to it all is that laboratories now have the capability to manufacture strands of genetic code whose purpose is to manufacture specific proteins. We know that genetic code makes proteins, right? The DNA creates proteins, and those proteins go on to regulate and to cause, to foster all the activities of a cell or of a human body.
Hey, our friend Elliot says that the Chinese developed vaccine for COVID does use a traditional method.
Yeah,
So
okay.
so that's in line with what Pasteur and some of these others have done, but you're going to be talking about a completely different way now, right?
Right. So because these laboratories now have this capability, and this was not developed in the last 7 months. This came out actually in about 2005. There was the first studies of how, well, I think in the 1990s, actually, they were able to do some of this, start to manufacture and produce genetic code. Some other things came on a little bit later, closer to 2005, but this is the development or the capacity to work with things that have been developed and have been under scientific study for a long, long time, right? And this Operation Warp Speed and all these different things that are coming out now are piggybacking off, standing on the shoulders of giants that allow us to come after this problem in a very effective way. It's called synthetic biology, one term for it. In other words, we can create the DNA structures that create and make the proteins that we want that DNA to make. So there's a lot of control over actions inside cells nowadays. And that capacity allows us to do amazing things. So that's the basic thing to remember about it, that as laboratories, we have this capacity. I wanted to introduce you to SARS-CoV-2 virus that's studded with spike proteins. You may have seen pictures similar to this. And it uses those spike proteins to alleviate, to allow it to combine with or move on to and enter human cells. Those spike proteins combine or match up with the proteins that are on the outside of human cells. If you see that little arrow in there, that's a gene, that's a representation of a gene. That is the gene that codes to make the protein, and that protein then makes those spikes on the outside of the cell. So that's
a little snippet of the viral DNA then.
Right, yeah. It's all a representation of all the different things that it has, but this virus can either have DNA or RNA. It can have one or the other. Some have DNA, some have RNA, but basically that's a gene on there that codes and tells that virus, or codes to make the protein to make that spike that's on the outside. Right. There may be a whole variety of spikes, but they're looking at a certain spike too, the S.
Yeah. Think of that as the teeth for the monstrous virus has teeth and claws, and that's the bit of genetic code of all of its genetic code, that's the little bit that encodes for the teeth and claws, as
I
it
think
were.
you're anthropomorphizing this a bit.
Oh,
So, there's two different styles of vaccines, and I'm not sure why that jumped to exactly what's there, but I'm was supposed to also bring in these others. I'm looking first at the AstraZeneca Oxford University vaccine. Those others are supposed to come up later, and I guess I didn't get that in my animation for this slide. But on the left side, this is a DNA-based model compared to the messenger RNA-based. The other two, the two that are out and approved right now, are messenger RNA-based.
the ones approved in the U.S.
The ones approved in the U.S., right? The AstraZeneca is a Swiss,
And Oxford lab,
and Oxford University is actually, UK. So that's their development. And they went with a DNA-based model, and we'll talk pretty carefully about what the benefits and drawbacks of each one are. It uses adenoviruses from chimpanzees. So chimpanzees aren't able to pass on their viruses we haven't had the contact that allows the adenoviruses that cause the sniffles and certain colds and flus in chimpanzees to activate in us. So it uses something that's not effective on us at all, and it takes this adenovirus, and then going back to the key of being able to transmit and implant certain parts of a genetic code inside this adenovirus.
it's kind of you talked earlier, I don't know, back in the fall maybe, about CRISPR, and you had these little lunar lander-shaped virus particles.
Yeah.
Right? And the adenoviruses are kind of like those little microscopic lunar landers that inject genetic code into your cells. But in this case, they've been hollowed out. All the guts have been ripped out of the lunar landers, and it's been replaced with this DNA.
Right. So this adenovirus, then, this happens to be its scientific abbreviation, which stands for chimpanzee adenovirus, and I think Oxford is involved with it, and then for the COVID-19. So it's a scientific structure that scientists can look at and understand what they're talking about and what this adenovirus has now been engineered to do. Now I want to get back to the other side. The BioNTech is a Swiss outfit, I think, and Moderna. These are both messenger RNA based, which we'll talk about more in a little bit. These RNA, are the messenger RNA are the detached codes that from when DNA splits apart in its replication process, it matches up, each one of those matches up with some messenger RNA that's in the cell, which then it codes for that, and that single strand can go off and create proteins as well too, or create more DNA.
It carries the messages of the genetic code from, it's a little bit of a simplification, but from the nucleus to the parts of the cell that actually synthesize proteins.
Which are ribosomes.
Right.
So these messenger RNAs are what is carrying the important message. The strength of that is, I guess, is that you now are working with what's a synthetically created messenger RNA that you can create to make any protein that you can code for, right? The weakness of it maybe is that RNA is much more fragile as a structure than DNA. DNA is combined and strong and caught in the double helix and is very strong. Messenger RNA deteriorates very, very easily, very, very easy to be attacked.
Just a hot or a warm water molecule zipping around it can break it apart.
Right.
So even at room temperature.
Right. So this modified synthetic nucleosides could both increase the protein production from the mRNA and drastically suppress the immune system's reaction. These were the two problems with the mRNA, that it didn't make enough protein quickly enough, and that the immune system in your body sees it as a foreign agent and can kill it very easily. So those are two problems, and this is what I was talking about, were both solved in 2005. The person that was one of the head scientists in 2005 that helped develop it is now one of the lead officials on the BioNTech group.
Right.
So,
Yeah, BioNTech. Yeah, it's a couple people online have said it's a German company.
is it? Okay.
Yeah. Not Swiss. They're very different.
Oh, yeah.
Right.
Okay. There was somebody that was Swedish. I don't know if that was AstraZeneca or.
Oh, Swedish. Okay.
Yeah.
Yeah.
So anyways, these were the two big problems. mRNA is very adept, easily made, much more easily made and created than the DNA, but it's much more fragile and much more susceptible to any damage. Like Scott said, if a warm water molecule could destroy it, imagine what the body's immune systems would do. So in 2005, there were ways developed to protect this thing, and they're called lipid nanoparticles. Lipids are fats, and they basically enclosed it for protection in a bubble of fatty molecules. So I know that works pretty well for me at times, on cold days and things like that.
Yeah,
I get protected.
I have a persistent bubble of fatty molecules.
So these lipid nanoparticles are a very critical part of the development of the capacity to protect the RNA and to get it to do its job. So we'll look further into that. But the two differences, I think I can go back and get the other ones in there as well too. And sorry for the little problem with the animations on this. So this is sort of like the lunar lander, like I showed in that other talk a while back. And this is actually, we're going to talk a little bit more at the beginning here. This is actually showing, if you're wanting to compare the two, this, you can see DNA in there. And so this is more representing the adenovirus and has DNA inside it. And that DNA is the gene for spike protein.
And that's it, right? That's pretty much the only DNA that this has been enlisted to carry around, is that spike protein DNA.
They've cleaned it out, basically, yeah. So it's an adenovirus that, and now you get into topics of alive and so on. It's not alive because it doesn't have all these different capabilities, but, and if I say alive in a few times on here, that's really not an accurate portrayal, but this is a virus that has the capacity not to reproduce really anymore, but it has the capacity to deliver what is in its inside the viral chambers.
This less fragile DNA,
Right.
it delivers it to the cells,
Right.
to your cells,
Right.
and mine, and
Yeah.
anybody who gets this particular vaccine.
So this is a next step. This is actually, you sent me something from the New York Times, Scott, and this was the development of how the AstraZeneca virus vaccine, works. And I thought that the pictures were very good, so I borrowed some of the pictures, and thanks to the New York Times for having much better artists than I am capable of being. But so now this is a section. Now this adenovirus has been injected into your arm, and it enters a cell. It bumps into some cells, and it is, because it's an adenovirus, and viruses are made to work their way into cells to provide their viral load into cells. That's what this still has the capability to do. So it comes to the side, its proteins get attracted to the side of the cell wall, and a bubble is formed and actually is encapsulated and brought into the cell. And this is where this is slightly different than what we'll talk about with the mRNA later, but these were the best pictures, and I thought we can kind of hopscotch off this and talk about the other ones from there. It deposits, you can see over on the left side of your view there, it's depositing a non-replicating DNA cell, or DNA gene, into the nucleus. That nucleus then knows through its replication processes and so on, it makes a messenger RNA from that.
The machine is the machine. It says, oh, there's some DNA that I can transcode right here.
Right. And it pushes it out of the nucleus, out into the cell.
But when it leaves, it's that messenger RNA. So it
Right.
translates it inside your cell.
You can see that when it's doubled, that's representing the double helix, and then it's the single is when it's messenger RNA. That messenger RNA goes and is then goes through its regular processes, the cell's regular processes. That's a ribosome up at the top there that looks like a copying machine or whatever, and it's reading, it's actually translating the code that's on the messenger RNA and making proteins from that. So as it reads that, it has the protein that's been developed in the laboratory to make a spike protein.
And that's it. No other part of the virus.
Right.
Just the spike protein.
Which is
That's really
not
important.
Yeah, right. It's a spike protein. It's nothing that's contagious, nothing that's of any kind of a pathogen, a problem. 3 of those spike proteins, and according to the New York Times, will make a spike, and those spikes and spike proteins and spike protein fragments are floating around in the cell, and they work their way out because that's their process of what the spikes do. They work their way to the cell wall and express themselves outside of the cell, and now you have these spike proteins and spike fragments that are outside the cell, giving the appearance of infection, although it's not infected.
It's a costume it wears that makes it look like the COVID
That's
virus.
a great way to say it, yeah.
Yeah,
So this is a vaccinated cell. It's not an infected cell.
right.
So on the right here, you see making antibodies. There's a vaccinated cell, and now the body's systems come along. That's a B cell in the middle there, and a helper T cell up above, and it comes along, and it's actuated by the helper T cell. The B cell comes along and finds these spikes there and starts interacting with them, and it comes along, and that's its job. It makes antibodies. Now, it's making antibodies for a disease that you don't really have. You just have the proteins that code for that disease or that are indicative of that disease.
The costume has fooled it.
Yeah, yeah. Good way to say it. I like that. So those are antibodies, those kind of Y-shaped things that are coming out of the B cell. And those antibodies, some stay on that B cell, and some get picked up by some killer T cells, and some just go floating through the bloodstream all around, and those things can then go on to anywhere that there's a spike protein that matches with it in your body, and they're there in flowing around for a while. As if you don't have the disease, those antibodies will die off like we saw in the video, but the memory cells are there that'll help make those antibodies again later on. So this vaccinated cell is engaging the whole immune system in your body to do this. Now, here's another virus cell that's gotten into your body, but your body has these antibodies all floating all around there, and they coat over those spike proteins, and they don't allow it, with that coating over that, they don't allow the virus to actuate and to get onto and get into an actual body cell.
So this is a diagram of an actual COVID virus particle,
A representation.
a representation of it, right? And it's decorated or costumed just like your cells were, but it's malicious, and your immune system has been pre-programmed to recognize its costume. So
Right.
it finds it.
Right.
Yeah.
And even if those antibodies aren't very prevalent in your system, the memory cells are there to see this, and very quickly, like it said in the video, it can take less than a day for these antibodies to be up in full strength and going after this and killing you before, or killing the viral cells before you even really feel sick. So, now this on the right is an infected cell, not a vaccinated cell, and it has those spike proteins because it's been infected by a real virus. But now these killer T cells come along, and they're assisted by other white blood cells in your body. Again, this would take a lot of time to go through and explain more about what each one does, and it's not critical for what we're talking about here. But this cell here is a killer T cell, and it is injecting a couple of different types of proteins into a cell that it's determined to be diseased, infected, and it kills that cell. And has the capacity to protect itself. This disease mechanism, or I'm sorry, the killer mechanism in itself doesn't kill it because it's protected itself, but it puts it into a cell that's not protected and kills cells that it determines that it's determined are infected with the viral load. So I hope that kind of clears things around. I wanted to talk about each one of the different Moderna vaccine, the Moderna vaccine, the Pfizer vaccine, and a little bit more because I think each one has their own strengths. And what I saw as I looked into it more deeply was that it's a little bit hard to say. You start reading about efficacy and 98% or 94.5% or different things. And it's actually, we'll see some things that maybe lead you to say each one has strengths and each one has more study to be done on it. We're at a very interesting time for this. So I have kind of a similar thing for each one. I just picked the Moderna vaccine, which again is a messenger RNA vaccine. And this is the protocol that went on. On this one, they determined that they were going to give you the vaccine on day 0 and day 28. If any symptoms occur 14 days after the second vaccination, the subject takes a nose swab. So of all the people that they tested with the Moderna vaccine, they kept records. They were in the tests and they kept records, and anyone that complained of symptoms was tested at the end. 30,000 participants. And of course, to do a proper scientific test, you need to split it between a control group, a placebo, and a vaccine group, okay? This is by the time you get to trial 3. The first trials, phase 1 trials, were on animals. Phase 2 trials are basically just developing the levels of vaccine to give to people and the safety of it and so on. And then you get to phase 3, which are more trying to determine results that are proper results for dealing with the virus. So this is in the phase 3 trial is what I'm basically talking about now. The control out of this group, the control group, 185 tested positive and 30 were severe. Out of 30,000 participants, split in half is about 15,000 people. So 185 that didn't get a vaccination at all, got a saline solution. 185 tested positive and 30 were severe cases. In the vaccine group, 11. So that's beautiful. 11 people out of 15,000 tested positive, and note, 0 severe cases out of this at all. So then they start doing efficacy, which you've heard a lot about, and I thought it's worth talking about a bit. When you talk about, you can talk about efficacy, which means how well it controls things, and you can talk about 2 different aspects here, and maybe more too, but one is against the disease itself, who caught the disease and who didn't. So you compare the groups, and the control group had 185 positive, and the vaccine group had only 11. So you take 185 minus 11, 174, and divide it by the total that had it in the control group, 185, and that gives you 94.5% efficacy. So when you heard the 94.5% efficacy, that was against the disease with the Moderna vaccine. It was 94.5% more effective compared to just the control. Okay? Then you can look at against the severe disease. Well, against the severe disease, 30 people got it in the control group and 0 got it in the other. So you take 30 minus 0 divided by 30, and that gets you 100% efficacy. So very wonderful results that can, against severe forms of the disease, and excellent results, much more than maybe any of us anticipated, against just the disease in itself. So efficacy can be measured in a variety of ways. Some of the things about it that you've probably heard: storage. It maintains viability as a disease, as a virus, as a vaccine, at -4 degrees Fahrenheit, which is about what your normal refrigerator, freezer would be able to do. 20 degrees centigrade below zero. So it can be transferred around fairly readily. Here's another important thing to think about. These are maybe a month old or so in these figures, but by the end of 2020, which we're at the end of 2020, Moderna said that they could provide 20 million doses.
20 million.
20 million doses, yeah. At the end of 2021, they said 1 billion doses.
Wow.
So they project that they can have 1 billion doses of this. Now, you need two tests or two applications of it, so that's 500 million people
You know, so
worldwide.
there are a couple of strange things, we may get into them later, about the efficacy of the virus, of the vaccine. I think this was specifically about the AstraZeneca one, that if they gave you a less highly
We'll talk
saturated.
about that, yeah.
Yeah, okay,
Yeah, it's an interesting thing.
yeah.
All right, so going on to the Moderna. Now, I kept this slide pretty well the same, just changed the title onto the Pfizer Oxford, and I just put in red any changes. So if you want to look at this, tested 0 in 28 days, 30,000 participants, how many, were positive in the control group and so on. I kept this pretty much the same, and I put in red the differences. Tested day 0 and day 21. Now, does that have a difference? Or you were given the vaccine 3 weeks after, not 4 weeks. And if any symptoms, 7 days after the second vaccination, not the 14 days in the other one, then they took a nose swab again. So these are people that reported a symptom, then would get a nose swab, and they were determined whether they were positive or not. I detect some problems with that.
Yeah.
Okay? All these things are just more things that, now that we know different things from these results, other tests should be run and can be run. 40,000 participants rather than 30,000, so that isn't significant to me. It's another good number at least, right? 162 tested positive in the control group and 9 were severe. In the vaccine group, 8 tested positive and 1 was severe. Okay? So again, it looks pretty good. It looks like something I'd put in my arm twice.
Yep.
Now, if you run the efficacy numbers, 162 minus 9, this is against, I'm sorry, that should be 162 minus 8 for the positive, right?
Yeah, it's
Yeah.
close, though.
Yeah. It came out again to 94.5% efficacy. And efficacy against severe disease really should be 9 minus 1. I crossed my numbers here when I was putting this together this morning, but it's very close anyways. It's about an 88% efficacy against severe disease as well. So very, very effective in that way as well, too, it seems like. But imagine, 20,000 people in the vaccine group, and only one of them got a severe reaction to this thing, right?
Got severe COVID or
Got
severe reaction to the
COVID,
Yeah,
severe
okay.
COVID, right.
Yeah.
Yeah, they're looking for COVID positive results from a nose swab
Right.
from people that reported that they had symptoms, okay? Storage. Now, this is the one that you've heard about. It maintains viability at minus 94 degrees Fahrenheit. Your refrigerator or freezer will not get to anywhere near that. That's 70 degrees Celsius negative. It's very, very cold. Very cold. You would have to have special freezers, special transportation systems, maybe better suited for use in an urban area where you can keep a whole store of it in one specific place. But again, a very effective vaccine. Again, an important question, the volume. By the end of 2020, they estimated they could have 50 million doses. The other was 20 million doses, I believe. At the end of 2021, 1.3 billion doses rather than 1 billion doses. So again, very effective, and if they use them properly in different areas, these could be effective, and the others can be effective as well. Finally, the AstraZeneca Oxford vaccine. This is the DNA version. Okay, let's see if there's some differences based on this. Now this one, they tested it on day 0 and day 28. And all participants took a nose swab 14 days after the second vaccination. So some of these, the other two, were based on you self-reporting that you felt symptoms. And if you felt symptoms, you were supposed to go back to your testing site and get a nose swab. Somebody may be non-symptomatic. They've talked about that a lot in this. Somebody else may be able to withstand something and say, I just had a sneeze or a sniffle or something like that. It wasn't really COVID or anything like that. And so this one tested everybody that was involved in the testing. All participants took a nose swab. 14 days.
So everybody was retested, not just the people who developed symptoms, right?
Right. Everybody was finally tested. Everybody was
Okay.
given a nose swab, all participants. In this method, there were 12,000 participants split between control and vaccine groups. The methods here get a little bit wilder, and we're going to have to spend a little bit of time talking about them. Scott already alluded to some of the issues. There was a test in Brazil in which there were 9,000 participants, roughly. Test in the UK in which there were 3,000 participants. Now, there's a variation in the test protocol, and Scott mentioned that. In Brazil, again, of the 9,000 participants in Brazil, roughly 4,500 were in the control group, got a placebo. They actually didn't get a placebo in this group. They got a vaccine for another type of flu. And so they wanted to have them kind of tested with something that may have been flu vaccine-like.
See if it helped.
It wasn't to help. It was.
Oh, I see.
It was to replace. Instead of getting the vaccine, they got the dose of this solution. The other cases, they gave a saline, a salt solution with no efficiency. This gave something that mimicked a vaccine
Okay.
to see if that had any impact at all.
Oh, I see.
Okay? So in the Brazil case, when they gave, in the 4,500 people that got the vaccine, they gave a dose of vaccine, and then they waited 28 days and gave a dose of the vaccine. Now, somebody misunderstood things or wrote things down wrong or something, because in the UK, the 1,500 participants that got the vaccine, the first day 0, they got improperly a half dose of the vaccine. They got half the volume of the vaccine.
It was an oops.
Yeah. And then the second time, on day 28, they all got a full dose. So there's a variation in here that needs to be noted.
AJ says there's another company in India that's developed its own vaccine. So there are several different
And
vaccines.
Russia has a vaccine that's DNA based, and China has a vaccine. So, again, with the AstraZeneca, the total results for all groups, when they combined Brazil and the UK together with all these variations, they had 131 people tested positive, and 0 people got it severely. Total results. So now they have a whole bunch of numbers to start working against, and I'm not going to do all the math up there. I did it already. But the efficacy against disease in Brazil seemed to be 62%, which maybe we're all turning our nose up against, 62%. But notice, in the UK, even though they only got a half dose for the first test, for the first vaccination, they had a 90% positive reaction, 90% efficacy. So that's a good number. And combined, because there were many more people in the Brazil test, it came to about a 70% combined efficacy rating, which doesn't compare with the 94s and 95s.
So,
But they had 100% efficacy against severe disease. Of the 12,000 people, 6,000 got the vaccine, and none of those 6,000 got the severe form of the COVID.
we're getting lots of questions online, so do you mind if I,
Let me just say one
Okay.
thing. The one thing I want to say is that this test, even though the efficacy numbers are low against disease, everybody got tested, right?
right,
So when you're sampling every person that took these tests, all 12,000 people split between control and Brazil and vaccine and UK, everybody of those 12,000 got tested, and nobody slipped through that had COVID but didn't present with symptoms.
okay.
So that would make me believe that probably you would have lesser numbers because you're testing everybody.
Right.
Whether they presented with symptoms or not, you would have lower efficacy numbers. So they can look lower here, but they aren't so bad. Go ahead, questions?
So, Mary asks if we know anything about how long the vaccine protection lasts.
That's probably one of the developmental issues that's going to have to go on, right?
We don't know yet, right?
Yeah, no one, because it's only been around for a while.
Yeah,
You know,
do the memory cells that it eventually makes in your immune system, do they persist indefinitely, or do they deteriorate or
And
go away?
that depends upon different viruses. Some viruses you need a booster shot for.
Right.
Some viruses you don't need a booster shot for. So this is something that's going to have to be developed with time. Hopefully, if we push down this pandemic to a point in which we're in pretty good control of it, those are the things you start sweeping up, cleaning up later when you develop as much immunization ability, as much, what do you call it?
Immune response.
Immune response in as many people as possible.
Right, yeah.
So, one final thing about the AstraZeneca Oxford DNA vaccine. Because it's DNA, it's much more robust. It's not as fragile as the RNA. It maintains its viability at 36 to 46 degrees Fahrenheit. So your regular refrigerator is at that temperature. You don't even have to freeze this stuff. You can put it in freezer boxes. You can put it in thermos jugs or thermoses and carry it to places. It's very good for portability, for the cost savings of having this stuff, and its viability under all these circumstances, maybe for developing countries, for rural areas,
Right.
for areas where there's less and less people. You don't have to
Yeah,
have
there've
a
been-
gigantic freezer or something like that.
There have been several stories I've seen that postulate that the AstraZeneca vaccine will be the one that heals the world. We may have the Moderna and the Pfizer vaccines that are effective in the developed world, having 5% or 10% of the world's population, but most of the world will be assisted and vaccinated because of the AstraZeneca vaccine and vaccines like it that are more robust and don't require the very low temperature for storage and whatnot.
Right. So here's the volume. The numbers I saw for that, the end of 2021, they are saying that 3 billion doses. So the others were 1 billion and 1.3 billion. This is 3 billion doses. Plus, when you look back at that thing, that maybe it's more effective with a half dose and the first dose, that could jump the numbers of doses up to 4 billion doses.
Right.
Which is significant, 4 times as many as the Moderna, for example. Okay? So, that pretty much goes through quite a lot of the different aspects of it. I did want to talk about side effects, and pretty interesting things there. There have been a very few minor reports of allergic reactions, which are always the case in anything that you put in your body. People are allergic to almost anything.
Yeah, any protein, any foreign protein in your body could cause histine cascade or other sorts of allergic reactions, but hypothetically, and given 7 billion people, somebody will have an allergy to nearly anything.
Right, and they've seen very limited, it is no different results than the flu vaccine that people go in and go to Walgreens at and so on. You know, it might affect different people, and there might be a tragedy or two or some, you know, or I'm saying a tragedy or two. I can't say what the numbers would be, obviously, but a very minor amount of those. And when you think of the tragedies we've been going through to get to this point, you know, it's something that ought to be able to be evaluated and probably accepted. Other than that, a lot of times the side effects are really things showing that the vaccine is really working. You know, that it's causing a response in your body will cause some of the pain and redness and fatigue, you know, and headaches, muscle joint pain. A lot of those things are there because the vaccine, is causing some type of immunoresponse inside your body. So if you go and you get a vaccine and you consider that maybe this little bit of redness and soreness or fatigue or whatever is just the small price you're paying for your body to be fully armed against a terrible killer,
Yeah.
You may be able to withstand it or accept it a little bit more too. All right. Well, I know we run full on our time like we always do, and appreciate people up top that are helping us out and so on. I do want to see if there's questions or concerns or thoughts. Hopefully this gave you some idea, and I really want to tell you that do not use this as medical advice. Don't take this as now you know everything there is to know about vaccines and viruses. Listen to what your doctors have to say.
So AJ asks a question, which I think is a great great question to prompt a follow-up that I wanted to make sure that we covered, Mike, and that is, can this technology for mRNA vaccines help with other sorts of vaccines, vaccinations, maybe pathogens, et cetera?
Yeah, definitely. In fact, they have been in use. One has come out already even before this. This has been an ongoing thing. One reason it came out in this 7 months was because there had been so much work done up till now. We were able to jump on this and use a technique that in the last 10, 15 years has really been emphasized. I think there's one out now for Ebola. That was out in March or something like that, had just come out with one for trials and tests. The other thing that allowed this to go so quickly was the vast amount of money that got thrown at it by the federal government and by governments from all over the world and by pharmaceutical companies and so on. So much money has been thrown at it, and a lot can be done when you have money, and money is no object to do this. But it's also being tested and developed, and one of the big effects before COVID-19 came around was the emphasis to do cancer, to fight cancer problems.
Yeah, apparently cancer cells have a kind of a specific protein envelope character compared to other cells in your body. So this mRNA and the DNA variant, this idea of only replicating the sort of the identity spikes on
COVID-19.
a pathogen, whether it be COVID-19 or any other pathogen, this idea of just replicating the little bit of that pathogen that triggers an immune response instead of a whole dead virus or a weakened virus particle. This is really revolutionary in medicine. It's a whole nother leap in humanity's ability to use technology and science to fight infectious diseases. So COVID-19 vaccination program is just the start of what will be a whole new era in science. These things can be developed, for regular vaccines, it can take a year or more. You were talking about chickens and chicken eggs and, you know, the laborious process that's required to make traditional vaccines is completely circumvented. These things can be built in a machine in a laboratory at a tremendous scale. So
Right.
just really amazing
A
stuff.
couple things that went on. Within a day of them identifying COVID-19 in Wuhan, China, they had the genome for this virus in one day.
Yeah,
So a lot of the other science that's been done about the Human Genome Project and all that stuff was brought to bear to solve this. The other thing is that this is so kind of cut and play kind of stuff that any step along this routine is so stable, so known that you can change an aspect of it to change it instead of going after COVID-19, to change it and go after Ebola or Zika or any other virus. And there's not, in the America, a lot of the regulatory regulations that cause you to have to prove so many things when you're dealing with proteins and building the proteins out of a pathogen and so on, you have to prove what you've been doing.
prove the
so on.
pathogen is dead, you know,
Right.
if you're going to use it in a vaccine.
I mean, and also prove itself to the Centers for Disease Control and the FDA,
Right.
the regulatory bodies. They just look at this and say, yeah, this is the gene for that, and you made this gene for Zika, or you made this gene for cancer, you made this gene for the proteins for really, for COVID-19 or COVID whatever when it comes
Right.
out. You know, so it's just a plug and play kind of thing. And regulatory rules were just completely sidestepped or not. They were eliminated. They weren't necessary. They weren't something. They were protecting.
Not
they were there to
applicable.
protect for something
Yeah.
that wasn't necessary in the case
Right.
of these devices.
Yeah, really just an absolute innovation in our
Yes.
ability to fight diseases. And another thing that I wanted to echo, you know, you covered it, Mike, but I want to make sure that our viewers hear this loudly and clearly. None of these vaccines reprogram your DNA, right?
Right.
This is not about genetic editing. You don't have to have any concerns about that. You know, we've talked about CRISPR and other genetic editing technologies in the past. You've done a great job of that, Mike, and that's not what this is. This is just programming your immune system with the minimal required amount of information to get it to respond to a particular pathogen.
Yeah. No, it's the more I looked into it, you know, the more excited I am about not only what it's doing for COVID-19, but what it's going to do for diseases and the health of the world, you know,
Yeah.
to come. So, you know, at least the silver lining in the whole COVID-19 cloud is that it may be generated such a vast wealth of emphasis and effort to try to overcome this, that it's going to be useful for years to come. People will survive things that they might not have because of what's been done with the scientists and lab technicians and our, you know, first responders
Yeah.
in that case, those types of cases that are doing that as well too. So,
So
yeah, it's fantastic opportunities.
I have another question online. Somebody says, are we sure? Who knows the long-term effects on our genome? And I really want to stress this point. The technology, the biotechnology, the way the molecular machines in your cells work, this does not have any potential to change your DNA. It's just, in order to change your DNA, you have to get into the nucleus of the cell and append something to your own chromosomes and what. It just, that's so far removed from any of the mechanisms of these vaccines. It's not possible. It's like getting from here to the moon in a rowboat. You know, it just, there's no vector, there's no mechanism for it to change your DNA. All it does is program some memory cells that exist that recognize particular external proteins on pathogen cells.
I think there's always a lot that can be said that good science means that you look carefully
Yeah.
at things, and we can't, you honestly have to say, you can't predict Well, you can't predict what long-term effects will be, but you need to monitor them and advise and investigate them and look at them and so on. But I think the rowboat to the moon is a pretty good analogy. There is not a way that you can say, oh, well, we didn't realize how this DNA could be altered from this or that. But, I'm not a scientist that can, be credentialed to talk about it much further than that, and it's well worth the investigation that goes into it. I think most people that are having anything to say about it are, that is negative, are naysayers and looking at,
They're not looking at it from a scientific point of view.
right.
They're letting their, emotions or particularly fear and lack of knowledge get in the way.
I mean, that could have stopped, somebody could have said to Edward Jenner in 1769, what are you giving this stuff to this kid for? Why are you, all right, so he probably won't get it. You gave him the cowpox, we'll give you that, but why are you putting smallpox in him now?
Right. I don't want to get horns on my head.
Right.
Right. But it never happened. Not ever. Right. People have been afraid of vaccination and medical technology as long as medical practitioners and from tribal healers all the way up to medieval medicine.
Sometimes with good reason, like I was talking
Right.
about George Washington.
He
at the
bled to
time it
death,
was-
right?
Yeah, he bled to death from people thinking that
Yeah.
they were helping him.
But this is one of the benefits of the modern scientific method, is figuring out real cause and effect and what the molecular mechanisms are behind these, and then manipulating them almost one molecule at a time. Really amazing stuff.
Yeah, yeah. It's fascinating. I really enjoyed my search into it, my look into it. Looking at each of those diseases, I would recommend, again, I think I do it every time we talk, some of these YouTube channels you get on to just type in AstraZeneca, Oxford, and the things they have to say about it. You can see some really, really knowledgeable people that are talking about some of the basic issues and basic portions of it. Just fascinating stuff.
All right.
All right, well, thanks a lot. I know Scott's going to be talking about some social sciences in the next time. I might put out something again on that same day, on the 17th. I'm going to be interviewing Steve Natz about the wisdom of the flock, and I think that's going to be his book
Launch? Yeah,
launch, yeah. And I might put something out on the email about that as well, too. He'd welcome you coming in and hearing what he has to say. To me, the interesting thing about this is the combination of science and some level of mesmerism, and what does it really mean to hypnotize somebody, and so that hasn't been fully decided and developed. And what Steve, a doctor, is trying to get at by saying that there's wisdom in science and there's wisdom in things that science, in his mind, can't fully explain yet.
Or hasn't yet, right?
And
Yeah.
hasn't yet, right.
All
Okay,
right.
thanks a lot, appreciate it, have a great 2021, we're happy to be here, and like I say, if you're interested in being part of the studio audience for the next one, send me an email, and we think we can probably squeeze about 5 people in different sections here, and have you be a studio audience that can give us a little bit more feedback and questions and so on too. So I think that'll be interesting development. So thanks a lot. Stay healthy. Get your vaccine if you're eligible, and stay safe.